My name is Sheri Ambriz, and I am the VP of Oligonucleotide Business Development at CPC Scientific. So thank you all for coming in so early this morning. Hopefully you've had your breakfast and coffee. I have not, so bear with me. But today I want to share with you some green approaches to the manufacturing of peptide APIs and the peptide-oligo conjugates. So the talk is organized roughly into about 3 sections. So we're going to look first at the introduction to POCs and green peptide chemistry. We're going to highlight the goals for making peptide manufacturing more sustainable. And then second, we'll look at our minimal protection strategies for the synthesis of 2 key peptides, and we'll look at those via case studies. And then third, look at some examples of some peptide-oligo conjugate carried out in more or less green solvent mixtures. So for those of you not familiar with peptide-oligo conjugates, I'll just give you a brief overview of their architecture. First, you have your targeting component, which consists of a peptide. So these peptides have a high specificity to cell surface receptors, such as like integrins, GPCRs, things that are often overexpressed in cancer cells. And then the peptide is attached to a linker, and this allows it to couple to the oligo. The chemistry of the linker is really important because it really determines the stability of the POC before it reaches the target, and also the mode in which then it separates from the peptide. And the peptide, the therapeutic component, in this case in oligo, can be encompass like single-stranded DNA, double-stranded DNA. It can do all of the standard therapeutic RNA oligos. All of these can be added siRNA, ASOs. We can also do morpholinos, make PMOs, PPMOs. And beyond the customization of the capabilities of these POCs, they also offer some exceptional therapeutic potential due to their enhanced tissue penetration, their high specificity and selectivity. They have reduced off-target effects, maybe some improved pharmacokinetics like extended half-lives, bioavailability, and then lower immune responses. So the chosen linker for a POC is really important for its intended functionality. Chemically cleavable linkers can broadly be classified into four main categories. First, you have the glutathione-sensitive ones, so you have the sulfide, the disulfide bonds. You have pH-sensitive linkers, as well as enzyme cleavable and photocleavable. The peptide-oligo conjugates are commonly associated mostly with the glutathione-sensitive linkers, so a disulfide bond is the most common, but also the enzyme cleavable linkers. So linker design can involve a broad range of different chemistries tailored to achieve very specific release mechanisms that you're looking for. So some of the linkers incorporate reversible functional groups, such as these disulfide bonds, which can really remain stable in their oxidative environment, but then cleave under their reductive conditions inside the cells. Other linkers, though irreversible, can be cleaved by intracellular enzymes. So in contrast, you have these non-reversible linkers, such as the triazoles that can be formed with click reactions or the thioether bonds. And these remain stable under most biological conditions, making them really useful for the applications where some prolonged stability might be required. So the peptide component of this conjugate is designed to target and bind specific receptors. And then through this receptor-mediated endocytosis, the entire conjugate can be taken into the cell and then contained in the acidic environment in the lysosome. So the linkage of this conjugation chemistry plays a key role in how the oligo is then released. Any premature POC cleavage by systemic enzymes can be a problem, as this will affect maybe the dosing or the overall off-target toxicities. So developing these POCs that undergo appropriate intracellular cleavage within the lysosome is crucial for the advancement of these POC drugs. So there's 2 main approaches to manufacturing POCs. You can either do a linear approach or a parallel approach. In the linear approach, peptide oligos are assembled just on the same solid support in a combined process. And there's 2 ways you can look at a linear approach. The first one is that you can have the attachment of a branched trifunctional linker straight onto the solid support. Then you follow that by the elongation of the peptide, and then you follow that by the oligo. The second approach, you can actually have the peptide elongated first, then functionalize that with a bifunctional linker, and then follow that by the oligosynthesis. But the linear approach has many constraints, as the chemistry for both the peptide and the oligo solid phase synthesis need to be compatible. And then the longer sequences you get, the more complicated the manufacturing can be. So in this parallel approach, which is the other way that we're doing this, is that the peptide and the oligo fragments are synthesized completely independently and concurrently, and then those fragments can be coupled together in a liquid phase procedure, followed by then the purification of the entire POC to yield the final product. So the parallel approach has the advantage of not requiring any compatibility of the solid phase peptide and oligo synthesis procedures. You can also accommodate longer peptides, longer oligos, and you have the diverse conjugation methods in order to be able to link these two. So you can really use the variety of this possibilities out there, depending on your functionality, the needs that you have. So the importance of using a parallel approach in POC assembly is that you can then get greener and more efficient pathways for the synthesis overall, because you can approach the optimization of each of these components independently. So the goal of developing greener manufacturing processes for solid phase synthesis is to enhance the atom economy and then reduce or eliminate the use of the environmentally hazardous solvents and reagents. So in the 12 Principles of Green Chemistry, which was introduced by Paul Anastas and John Warner, which provides a framework for creating safer and more sustainable chemical processes. The principles focus on reducing the waste, minimizing the use of hazardous materials, improving energy efficiency, and incorporating renewable resources. The goal is to prevent pollution at its source rather than just dealing with it after it's created. So the principles are critical because they allow the industry then to reduce their environmental impact, improve their safety, achieve long-term sustainability. And by following these guidelines, companies can then reduce their carbon footprint. So we're going to continue to innovate and deliver high-performing products. So in alignment with these 12 principles, we're going to really focus here today on using less hazardous chemical synthesis, so whenever possible, selecting safer solvents by avoiding any unnecessary reactions, and then choosing those processes which are compatible with eco-friendly solvents. And then third, most importantly, we're going to avoid any unnecessary derivatization, such as the use of blocking groups. So this protection, deprotection steps, all of these temporary modifications, they're physical and chemical processes, generate a lot of waste and then decrease the atom economy of the overall process. So for the success of green chemistry, these solutions need to be technological, they need to be innovative, we need to have safer chemical designs. Natural doesn't always equal sustainable, so these, all these natural materials are not necessarily environmentally friendly or efficient for the process. So the products need to be efficiently manufactured, they need to be cost effective and safe and non-toxic. So not only do we want to minimize the waste in materials, but we'd also like to minimize the waste in energy. So we want to avoid excessive heating, any long reaction times, and remove any of these unnecessary steps. We also want to maximize the material use, which really means that all of the inputs into the process should ultimately be incorporated into the final product. So we want to favor those reactions that avoid any extra reagents, protecting groups, catalysts, things of that nature. So in solid phase peptide synthesis, these amino acids are sequentially added to a growing peptide chain anchored to a solid support resin. But after the coupling of each in the deprotection step, this resin-bound peptide undergoes washes to remove all of these excess reagents and byproducts created. So solid phase does offer key advantages over liquid phase. So the use of these excessive reagents actually drive these reactions to completion, ensuring a more complete and efficient couplings. But also these synthesis steps are carried out in the same reaction vessel, so you eliminate the need for any kind of material transfers. So as a result of all of this, these intermediates that are created do not require any purification, isolation, characterization, until this final product is completed and cleaved. But despite the advantages of solid phase, during this elongation phase, you have this repeated washing, usually something like DMF, in these filling, draining cycles, and it's required, this heavy washing is required to remove any excess reagents and prevent any contamination further down the road. So this effective washing is really crucial for maintaining high yields, minimizing side reactions, particularly during these longer synthesis, but this creates a lot of waste. So synthetic peptide formation is very energy efficient to make a single amide bond here. You have your peptide dimers, you start with 2 amino acids, and in order to do that, multiple reagents and protecting groups are required to be in this process that are not ultimately incorporated into your final product. And so as you can see here, all these groups in the bottom will all be discarded in the process. So here you can really visualize the waste in the process. So the main chain protection during peptide elongation is typically achieved using FMOC chemistry. And over here you can see that in blue. The FMOC protected amino acids are then subsequently coupled with an activation at the C-terminus using a coupling reagent, also in blue here. So the FMOC is removed under basic conditions, typically with something like piperidine, and then the resulting byproducts are then washed away. So in certain amino acids, they require side chain protections as well, and so you can see those here in red. And those ones, all those side chains reactions during the chain elongation or the cleavage from the resin, those ones all need to be removed as well. So all of these added protection groups, the reagents in blue and red in this process, are all going to be ultimately washed away and never even incorporated into the final peptide. So here you can just see a real quick cartoon of how nature approaches this very elegantly. During the translation, the mRNA read by the ribosome, the transfer RNAs, they bring that corresponding amino acids, build in your peptide chain, and each of these tRNA matches with the specific mRNA codon, ensuring that this precise assembly of this peptide. The process remarkably efficient, sustainable, no materials are wasted. Both the ribosome, the tRNA then are reused in multiple rounds of the protein synthesis. So we can only hope that in synthetic synthesis we can get to this level of efficiency at some point. So this is a solvent selection guide, and so it really helps identify reagents to avoid or try to reduce in your process. The removal of these protection groups really requires large volumes of trifluoroacetic acid, of the TFA, which can result in very lower yields. It actually poses a very significant risk to the environment as well. So while the use of some of these side chain protections reduces common side reactions in SPPS, the washing away of those protecting groups can actually require significant effort just to avoid the reattachment elsewhere on the peptide that then may become permanent and compromise the crude purity. So the solvents commonly used in SPSS are kind of spread among all levels of this solvent selection guide, but you can see in bold here the ones that are really highlighted, the diethyl ether, DMF, DCM, ACN, so you have acetic acid and water. These reagents really vary significantly in their environmental toxicities. So our goal is to eliminate the protecting groups, i.e. with a minimal protection strategy that can require highly hazardous reagents in order to remove those, such as the diethyl ether and TFA. So while the use of other hazardous solvents like DMF can be reduced, it can't always be eliminated, but there is ongoing work, of course, to replace that. So the objective of this work is really to introduce recommended solvents whenever possible, and then you'll see this later, but particularly we would really like to look at those reactions that are compatible with aqueous solvent mixtures. So if we use the parallel approach to this peptide-oligo the peptide-oligo conjugate assembly, then we can actually address the improvement of each of these components independently, maximizing our overall benefit. So for sustainable peptide manufacturing, we're going to look at this minimal protection strategies. And as mentioned earlier, some of these side chains of the amino acid really require temporary protection groups in order to avoid any kind of side reactions. There are some of these amino acids that don't require any protection at all, so we'll take a look at these real quick. We're going to look at both of these. We're going to showcase this with a couple of case studies where the peptide elongation is achieved using unprotected side chains. We have serine, threonine, tyrosine, so you can see up above the list of all of those. The strategies are going to be applied to two specific peptides. The first one is a novel peptide small molecule conjugate, which incorporates this acid-sensitive small molecule. And the second one is a goserelin peptide, but we're actually going to look at the API impurity is what we were synthesizing. So the goal here is really to demonstrate how minimal protection approaches can simplify the synthesis, but while still maintaining the efficiency and preserving some of these sensitive functional groups. So certain amino acid side chains require protection due to the presence of the strong nucleophiles or electrophiles. The amino acid with the nucleophilic amino groups, such as like lysine and ornithine, are typically protected with TBOC. And this needs a strong reacid to remove it, so up to say 50% TFA to remove that. The side chains with the electrophilic carboxylic acid groups, such as the aspartic and glutamic acids, are protected with the terp-butyl groups, and those can be achieved using even a stronger acid like 95-100% TFA. And those often then require scavengers as well, something like water or phenol. The cysteine, with its thiol group, is also protected with a tridal, and that also requires 95, 100% TFA to remove. And then you have this aliphylic amino acid. So this group on the bottom, that group on the bottom there doesn't actually, they have no reactive functional groups, so they don't actually require any side chain protections. And then you have an entire group, which is sort of what our focus would be on today, of amino acids that have intermediate reactivity. So those ones can often be left unprotected, but under the appropriate synthesis conditions and cleavage conditions. So in traditional SPPS, the hydroxyl groups of the serine, threonine, and tyrosine chains are protected with T-butyl groups, which, like I said, are removed during cleavage with 95 to 100% TFA and some scavengers. The T-bot group on the tryptophan and the tridal groups, those ones can be removed similarly. Arginine's a little more complex. The protection strategies have evolved to minimize some of these cleavage times and reduce the side reactions so that the prolonged exposure to these acids actually can degrade the peptide. So here you can see the conventional strategy with all of the protecting groups. You can see these shown in red here. So the elongation is carried out on solid support using the appropriately FMOC protected amino acid derivatives and then a coupling reagent. So the cleavage is then performed using a TFA cleavage in a 95% cocktail, as I'd said. But this is then followed by a precipitation of the crude peptide with cold diethyl ether. Then the crude material must be extensively washed with this cold diethyl ether, in this case, for a total wash volume of 350 liters per mole. So if you remember from the previous slide, the solvent selection guide, that the diethyl ether is actually in the worst category, in the highly hazardous category. So it is definitely one that we want to reduce or eliminate. So with our minimal protection strategies, you can see that all of those protecting groups are no longer there. So nearly all the amino acids are left unprotected. The hydroxyl groups are weak nucleophiles, so they don't really interfere with the coupling of other amino acids. The exception here is lysine, so it is protected here still with the labile DDE group, and this can be removed with very dilute hydrazine versus a very concentrated TFA. So the peptide cleavage from the resin is achieved using this 30% HFIP in dichloromethane, but acetic acid can be used as an alternative as well. So this approach eliminates the use of both TFA and diethyl ether in the process altogether. So in this side-by-side comparison, you can actually observe that the amino acids that require the side chain protection in the conventional approach on the left, and in contrast, you have the minimal protection approach on the right. And this cleavage step now, if we've removed these protecting groups, this cleavage step can be altered to a more favorable process. So the conventional approach uses the TFA with scavengers and large amounts of diethyl ether, while in the minimal protection strategy, it employs only a 2% hydrazine, HFIP, and DCM. It doesn't require diethyl ether or TFA at all. So in the large-scale comparison, you can actually see the benefits of minimal protection. They're pretty obvious here, is that the quantities of the TFA and diethyl ether are considerable. Combined total of both of those reagents is 380 liters per mole for this traditional approach. And then the reagents are eliminated completely from the process in the minimal protection approach. In addition, the amount of solvent goes from 380 liters down to only 22 liters in the new process. And the atom economy is actually improved with the elimination of these protecting groups. So the crude purity here you can see on the bottom, that can actually be a little bit misleading. It's mainly influenced by the acid sensitivity of this small molecule that's in the conjugate. But this is another reason then why we would want a different approach to producing this peptide, especially when conjugates, other things are incorporated into the peptides. So as mentioned earlier, the small molecule in that conjugate, you can see here, degrades under acidic conditions, so which is reflected for these two large impurities on the chromatogram on the left. But aside from that small molecule-related impurities, both of these spectra show similar crude profiles of the main product. So the elimination of these protecting groups didn't negatively impact overall the purity of this crude oligo. So in part 2 of the minimal protection, we expand the approach to include some more difficult amino acids. So here we include the unprotected side chains of the histidine, tryptophan, and the arginine. So we demonstrate here the synthesis of the goserelin peptide API impurity. and this one is actually it shows like a convergent approach with a fragment strategy. And so we can actually still eliminate these side chain protections. The only one here is that similar to kind of other peptides in this category, like buserelin, we do have the D-serine actually does remain protected with the T-butyl, and that is incorporated into the final product and remains throughout the synthesis. So in this depiction, you can see here the synthesis scheme. You can see that there are two fragments. There's a 7-mer fragment and a dimer. And the dimer remains bound to the resin support for the coupling of these fragments. So in this convergent approach, we actually are combining a 7-mer fragment, which contains the T-butyl protected serine, with the resin-bound dimer fragment. This is at the C-terminus, and then the coupling of this, the peptide is then cleaved after the coupling from the resin to yield this goserelin impurity. So this is just a really quick, high level to show that we have developed several different approaches to looking at minimal protection and to improve these processes to eliminate these reagents. So I went through those really, really fast. If you guys are interested in much more detail in the chemistry behind that, we do have white papers on this minimal protection strategy. You can go to our website, you can download that. There's a QR code here if that works on your phones as you're taking pictures. So the key takeaways from the minimal protection approach of the peptide are that the principles of green chemistry have enabled for the efficient and the sustainable synthesis of both the peptide drug conjugate and this goserelin API impurity. So we can show an increase in their atom economies by the reduction of these unnecessary protecting groups. Specifically, we have the T-butyl, the PBF tridyl, and the TBOC. And we also utilize safer solvents by the exclusion and the replacement of the diethyl ether and the TFA. And then we significantly reduce the overall volume of reagents for the entire process. So once we have the peptide manufactured using green techniques, we can then move to the assembly of the full POC. So as a quick reminder here, the slide I showed before, there are several possible linkers depending on the functionality and the needs of your application, and some of these processes can be more green than others. So some of the linker conjugations are already relatively green, and they have not and do not need to be altered. So these sulfide linkages can actually be performed using several methods, modifying the oligo component with various linkers and then coupling that direct to a cysteine. So this can either be non-reversible or reversible based on the linker chosen. There are also some advantages and disadvantages to each that really depends on the functionality of what you're trying to do with this peptide-oligo conjugates. The thiol here can be very efficient. This reaction is passive, and it can be formed in just water and other safe reagents, resulting in very high yields. The next one here is the malamide conjugations. These can be performed via the amino-modified 5' end of the oligo. This can be synthesized directly by solid phase synthesis on the oligo, and then that amino group is functionalized with a malamide group in liquid phase using something like an NHS ester. And then you can follow that to the conjugation of that free amine on the end terminus of the peptide. So this process also can be very efficient in atom economy. So most of this functional group is then incorporated into the final molecule, and then no additional reagents are really needed for this reaction to occur. So you can see from the images to the right here that the final product here is very clean. So the click chemistry reaction, specifically we have this copper catalyzed azide alkyne cycloaddition, or just called the click reaction, offers the significant advantages over the traditional Husgen 1,3 dipolar cycloaddition. So this is really a big improvement over the standard process that was done many, many years ago. So while the original reaction actually forms a mixture of these isomers under thermal conditions, the click chemistry can actually provide a highly selective, fast, and efficient synthesis with a single isomer. So the use of the copper catalyst in the click chemistry actually drastically reduces the reaction time and enhances these product yields. It makes this process much more practical and versatile for a wide range of applications. Additionally, this click chemistry is compatible with aqueous conditions as well. And then a variety of other functional groups, offering really good flexibility and the biocompatibility compared to this uncatalyzed Husgen reaction. So we really do encourage and promote the use of click chemistry, mostly because of its orthogonality to the other methods of the peptide chemistry. We have its modularity that enables this convergent synthesis, and its compatibility with green chemistry principles and eco-friendly solvents. But if you do want copper-free, so we have a copper-free offering as well. So typically POCs are prepared with the alkynes on the oligo and the azides on the peptide, though those functionalities really can be swapped if needed. The standard alkyne group does require a copper catalyst to facilitate this click reaction. However, if we use strained alkynes such as DBCO or BCN, that will allow for a copper-free conjugation. And notably in this copper-free, this can be employed not only to eliminate the need for copper, but it also enhances the reaction rates due to this kinetic benefits of this ring strain. So whether we use the traditional copper catalyzed or we use copper-free conditions, the reaction actually can be performed in environmentally friendly solvents such as water or ethanol. So a range of these strained alkynes can be used for the click conjugations of the peptide-oligo conjugations. So we have the BCN, which is very advantageous for fast reaction rates, while the DBCO is actually more stable under these physiological conditions, so making it more of a common reagent in the bioorthogonal chemistry. The slower reaction rate, the DBCO, also offers like more controlled conjugation possibilities, which is critical for applications for like targeted therapies or drug delivery systems. The slower rate can offer some greater control, some greater precision, particularly in the biological and the therapeutic applications. So the scheme on the right here, this just simply illustrates our use of the DBCO PEG NHS esters. So this is incorporated into the amino-modified 5' end of the oligo. And the alkyne group of the DBCO then reacts with the azide on the lysine chain of the peptide. So the incorporation of the alkynes into the oligo portion of the POCs can actually be efficiently achieved through phosphoramidite chemistry on the oligo. So the method allows for the addition of the alkyne groups at either the 5' end of the oligo or internally as maybe a modified base, providing great flexibility in the design of how you make your POCs. We're also able to make many of these modifications on the 3' end as well, but that can kind of be, usually those reagents need to be a little bit more custom, and that would just add both time and resources to the development. But once incorporated, these alkynes can participate in the copper catalyzed or the copper-free click chemistry, both of which are really widely used for their specificity and their efficiency in forming these stable covalent bonds between the oligo and the peptide. So a lot of what has been learned from the development of antibody drug conjugates can actually be applied to these peptide-oligo conjugates as well. So here you can just see some examples of cleavable linkers that can be used for some specific enzymatic cleavage. So in 2004, Lobov and coworkers introduced this valine citrulline, or VC-PABC linker. So this sequence is actually a substrate for the lysosomal proteases, which can be overexpressed in cancer cells. So it makes this linker actually a really good option for something like oncology applications. Once the POC is internalized into these cancer cells via endocytosis, then it can be transported to the lysosome where the linker is then cleaved, releasing its payload. So the choice of linker really allows for the cleavage at both places on the molecule as well as places within the cell. So here the importance of using this modular approach to actually putting together POCs. It allows optimization for each of these components and the flexibility to use various modifications as needed in your final functionality. So if you can see on this molecule got lots of things incorporated. You can add the BCN using green chemistries. You can incorporate this VC-PABC cleavable linker, as well as any other modifications. You can see in the lower corner there's a 6-FAM. So you can add fluorescent dyes. So the possibilities are numerous. So to ensure this optimal and the complete conjugation of the peptide and the oligo that results in the formation of your final POC as you want, it's really crucial to monitor this reaction. And so we've developed analytical reverse phase techniques that can actually look at this entire reaction and watch it progress. So this really allows for the confirmation that we have the complete consumption of the limiting reagent, and in this case it was the peptide. So any excess oligo can then be recovered and subsequently used. So this really ensures the most efficient conjugation possible and eliminates the wasted components in the process. So not only does a parallel POC synthesis allow us to synthesize peptides and oligos concurrently and significantly reduce the turnaround times, but it also allows for the independent of each of those methods to be developed. So here you can just see a standard peptide. What this might look like is that you have the oligo and peptide produced probably within about 2 weeks. This is for obviously very large scale, and it depends on the length. But the conjugation can then occur in that 3rd week, conjugation purification. So, realistically, we have a standard peptide oligo, fairly simplistic. This whole thing can realistically be completed in about 4 weeks. So here are some of the key takeaways here for the POC, and using this parallel approach to POC manufacture really has the advantage of not requiring any compatibility of your solid phase peptide synthesis and your oligo solid phase synthesis. And you have the accommodation of longer peptide and oligo fragments, and you have the availability to do these diverse conjugation methods to really tailor it to your application. So this leads to the ability to develop greener and more efficient paths for the synthesis of all of these components.